All right, last one for this, this lab. So, last one on back of page 58. We've got something called sex linkage, which is a little bit weird, and I'm going to try to explain it as well as I can. So, there are things called sex-linked traits. Now, if you guys remember, or if you know, sex-linked traits means that they're on the sex chromosomes. So the XX or the XY chromosomes. So like this. The thing about these is, X chromosomes, they're the same size. However, in the real world, the X chromosome and the Y chromosome are very, very different sizes. The Y chromosome is actually very, very small. And because of this, we have some problems. There are a lot of genes on the X chromosome. Because the Y chromosome is so small, this X chromosome, or any X chromosome, in anybody, this does not have an analogous gene on the opposite chromosome, which means if we were talking about any given gene, A, B, whatever, everybody's got 2 copies, right? Because you get 1 copy from mom, 1 copy from dad. In this case, we don't get that. You might only get 1 copy of each for this particular chromosome because the Y doesn't have it. The Y is so small in comparison, it can't hold very many genes. Hardly any at all, in fact. So what we end up having is the genes that you get from the X are the ones that show up. And if those genes have a mutation that gives some sort of disease, you're going to get the disease. So, there is a disease that's sex-linked called hemophilia. And hemophilia is an issue with blood clotting. So there are different types of hemophilia, but basically when you get a cut, you get an injury, your body sends lots of proteins and other things to that area to help clean it up, to help make sure that your blood clots and makes a scab correctly. However, people with hemophilia don't have the clotting factors that they're supposed to in their blood. So their blood just runs and runs and runs, and it doesn't clot, so it doesn't stop. And especially in the past when we didn't have things like blood transfusions and things like that, people died from hemophilia fairly frequently because they couldn't even get mildly injured. A bruise could kill them because their body just wouldn't stop bleeding. So that's the example we're going to use. So let's say we have these 2 adults. We have our female and our male. Now, hemophilia is what's called a sex-linked recessive gene. And that just means it's recessive, but it's on the X chromosome. So I'm going to write out what that means. So, okay, so the way we represent this is, again, using the subscripts, but this time we're using letters instead of numbers. So, dominant, capital letter. So we're saying that this X chromosome has the dominant copy of that particular clotting gene. Now, in this disease, the dominant is the normal one. The dominant one means that your clotting factors are normal. They're perfectly fine. You can bleed, you can make scabs, you're fine. This one, however, you're going to have some problems. So this is an X chromosome that has the mutated gene for hemophilia. So if an individual, let's say this adult male, has this gene and has a Y that doesn't have an analogous gene on it because the Y is so small, this guy if he was like this, he would have hemophilia. So now if someone, the example in this is, a woman and a man. The man, in this example, the man is normal. So he has a normal clotting gene and of course a Y chromosome. We have a woman who has one normal gene and one mutated gene. Now, if you've got somebody like this that doesn't have the disease itself, but has a gene that could pass it on, the word that we use for that is carrier. So this woman is a carrier of the hemophilia gene. So let's say these two are going to have a baby. Now, if these two have a baby, for mom, there are two different things, of course, that she could pass on to her kid. Mom could pass on the big H or the little h, because remember, these have equal chances of being passed on. Dad could pass on the big H or the Y. So if you have a Y, you're male. If you have a big H, in this case, on the X, you'd be female. So we can draw out a Punnett square for this. And this is what our Punnett square is going to look like. So mom's across the top. We have her big H and her little h because she's a carrier. We have dad down the side. He's got a normal gene and the Y. So let me write everything out. So it's kind of hard to read because it's a marker, but so we've got a few different things here. This one in the upper, over here in this upper corner, is big X, big X. So this individual is homozygous dominant. So this individual does not have hemophilia, and because they're XX, they're female. So we have a female with no hemophilia. So female, no hemophilia. Up here, we have an individual who's big H, little h. So they are heterozygous for the hemophilia gene. They don't have hemophilia because the big H is the dominant one, that's the normal one, but they're a carrier. Wait. Yes, that's an X. It looks like a Y, but it's an X. That's a little bit better. So again, big H, little h is a carrier, but doesn't have the disease, and because they're big X, or XX, they're female. So we have one female without hemophilia at all. We have one female without hemophilia, but is a carrier for the gene. That's what the little star means. And down here, we have this guy. We have a XY individual, so this individual is male, and this male has a big H, meaning that they do not have hemophilia, and they can't be a carrier because they don't have a second version of that gene. So we just have a male that's got no hemophilia. Male, no hemophilia. Now this one, down here. This individual is another XY, so we have another male, But this guy has got the lowercase h, and that means he has hemophilia. So we have 2 females without hemophilia, one is a carrier, 1 male without hemophilia, and 1 male with hemophilia. So these guys have a 1 in 4 chance of getting a child with hemophilia, and that gives us a 50% chance of getting a son with hemophilia. Next example we're going to do is we're going to look at color blindness. Again, another sex-linked trait, but we have a slightly different situation this time. All right. So here's what we're doing. We've got a family. We've got a mom and a dad. We've got the main character, Nancy, and she has a sister and a brother. Now, Nancy's dad is colorblind. We're using C. Capital C is dominant normal vision. Lowercase c is colorblind. So dad's colorblind, so he's got a Y because he's male, and he has to have the lowercase c for colorblindness. Nancy's mom has normal vision. Nancy's sister is colorblind. And Nancy's brother has normal vision. So we're going to use this information to try to figure out what the genotypes are of the sister, the mom, and Nancy. Because we don't know. We don't know what those other X's are. They could be big C, they could be little c. We don't know, so we're going to try to figure it out. And the easiest way we can do to figure it out is to do a test cross between mom and dad. We know what dad's genotype is. Dad's genotype is X, little c, Y, because dad's male and he has color blindness. So, looking at Nancy's mom, we have 2 different options. Nancy's mom has normal vision, so she can either be big C, or big C, little c. So what we can do is write out our Punnett squares to figure out which one it is. For the sake of brevity and stuff, I'm just going to write the lowercase letters. hopefully abbreviating that like that makes sense because I don't have a lot of room on the board. So over here on this side, we have, if we're guessing that mom is big C, so she's X big C, meaning that she's got 2 normal copies of the gene, which means that she does not have color blindness. Dad, we know, is X little c, Y, because he is colorblind. So if mom's big C, then this is the spread that we get. This individual is female, not colorblind, but a carrier. This individual is female and colorblind because there's 2 recessives, 1 from each parent. This individual is a big C, Y, so this is male with normal vision. This one would be also a big C, Y, male with normal vision. So in this case, all the males are going to have normal vision. Up here, we have a 50-50 chance of getting a female with normal vision and a female with colorblind vision. Over here, it's a little bit different. Again, we have dad. We know dad's colorblind. Mom, we're trying to figure her out. So if we've got 4 different things here. So the first one right here, big C, little c. We get the little c from dad, big C from mom. This individual is going to have normal vision, but be a carrier. This individual is going to be female and be colorblind. This individual is male because he has a Y, and he has a capital C, meaning that he's a male with normal vision. But over here, we have our Y, so this one is also male, but a lowercase c from here, so this is a male with colorblind vision. So if we look at these, we see we've got females here, Nancy mom, sister. What do you guys think? We know brother is big C, Y. So technically speaking, I made a brief error there. This one is supposed to be big C, little c. That's where I was messed up. Sorry. You probably caught that and were yelling at me through the computer. It's fine. So, yeah, big C, little c over here. So, in this case, you know, Nancy mom, sister. Sister's going to be the key here to figuring this out. Because Nancy's sister is colorblind. Meaning that Nancy's sister has to be little c, little c. So if Nancy's sister is little c, there's only one of these 2 options that could possibly be the case for mom, and it's this one, because this is the only situation that gives us a female that is colorblind. So mom has to be big C, little c. So this is our case. And if we know that mom's big C, little c, we can figure out what Nancy is, because Nancy has a big C. And the only situation here in this box where Nancy could have a big C is if she also has a little c. So here's our family. Nancy is big C, little c. Mom's big C, little c. Dad's little c. We have 2 little c's for sister and a big C for brother. And that's it.